Hello students, welcome back to our channel. Today we are going to talk about herbicide resistance. As you know, this is very important topic from your exam perspective. Whether you are appearing your semester exam or any competitive exam, questions generally come from this section in GRF, SRF as well as NET exam as well as ARS exam also. So let's begin. So what is herbicide resistance? According to, there are two definitions proposed by Weed Science Society of America and Herbicide Resistance Action Committee. First one is Weed Science Society of America proposed that herbicide resistance is the inherent ability of a plant or biotype to survive and reproduce following exposure to a dose of herbicide normally lethal to the wild type. For example, if we are applying any herbicide like 500 ml per hectare, if we are applying, and that is capable enough to control that particular weed, but after few years, if we are applying even that 500 ml per hectare and that is not capable to control that weed, then we must assume that there is certain resistance developed during this period, during this gap. So that is basically what herbicide resistance means. So due to some genetic change or due to some alteration in genetic factor, so there may be some mechanism has been developed by the weed which can resist to that particular herbicide application at a particular dose that is being controlled earlier, but it cannot be controlled at that particular dose. So that means it has developed some kind of resistance. So this classification was proposed by Weed Science Society of America. Next is Herbicide Resistance Action Committee proposed herbicide resistance as naturally occurring inheritable ability of some weed biotype with a population to survive a herbicide treatment that would normally control that weed population. So basically what according to HRSC, what was the definition? That on application of herbicide treatment at a particular dose, it is earlier controlling that weed population, but due to some natural phenomena, some inherited ability has been developed in that particular weed flora, so that it can survive to that herbicide application. So this is basically herbicide resistance. So first proof of herbicide, it was initially the first wild carrot has been being developed, the resistance to 2,4-D, and it was reported in Canada in 1963, but it was not highlighted at that time. So the first highlighted report of herbicide resistance was a common ground cell, that is Senecio vulgaris. Senecio vulgaris. It has been developed resistance to atrazine herbicide, and it was reported in Washington state in 1970. Okay, so it was highlighted, this common ground cell, that is Senecio vulgaris has developed resistance to which herbicide? Atrazine, that is one herbicide from triazine group. So this atrazine herbicide, it is resistance, this weed shows resistance to atrazine herbicide, and it was reported in 1970. Next, development of herbicide resistance. How herbicide resistance developed? There are certain factors which carry this development of resistance, which are responsible for this development of herbicide. If we use the specific herbicide again and again, repeated use of specific herbicide or combination of herbicide, if we are using a single herbicide or combination of herbicide again and again, then there may be a chance of development of herbicide resistance. Then weed population with a wide genetic diversity may develop resistance rapidly, especially for herbicide with a single mechanism of action. If we are using the herbicide with single mechanism of action, then the development of herbicide resistance, it will be rapid, because the action site is same. If there is any modification in that action site, then that particular weed is capable to survive to that particular herbicide application. So again, that is some kind of genetic change or some kind of mutation happened inside that particular weed, so it is capable to survive in that herbicide application. So again, that is genetic. So when it produces certain seed, and after few generation, those seeds will be resist to the particular herbicide activity, as this is genetic, right? So in that case, if we are using single mechanism herbicide, single mechanism of action herbicide, then the chance of herbicide resistance is more rapid. Then weed possessing characteristics like large plant numbers, prolific seed production, high rate of weed migration, and diverse environmental condition may contribute to the high genetic diversity, right? Next is, where do resistance weed come from? कहाँ से means from where this resistance will come from. So initially, if we are applying, whenever, suppose this is our crop field and these are the weeds, and we have applied the herbicide on the foliage, right? So one plant, due to some mutation or maybe genetic change or maybe any type of change, suppose this plant developed resistant to that particular herbicide. So this we applied the herbicide all over the crop field. So, and from that particular weed population, this particular weed showed resistant to that particular herbicide. Next, this weed plant, it survived to that herbicide application. Then what will happen? In the next year, it will produce some seed. This weed plant, it will produce some seed. So again, in next season, if we are applying certain herbicide, that particular herbicide, then few of these weed will survive because they are resistant. They have developed resistance. Then after few season or after few year, all that species has developed resistance. So all these weed species, that particular species, can be survive to that particular herbicide application. So there we will just assume that this particular weed has developed resistance to that particular herbicide, right? Then next thing is, if there is any DNA mutation, as you know, mutation is rare. That means in a million, maybe one plant get some mutation, some kind of mutation due to different environmental factors, due to some exposure to different radiation. So if any changes in that DNA, particular DNA level, if mutation happens, and we have applied herbicide, so that plant got survived. Okay, among these, there are complex flora of weeds are present in the field. So one weed is survived. Okay, then again, if this weed survived to that particular herbicide as it has been mutated, so after it produce some seed, after flowering stage, if it is producing some seed, those seed will also become herbicide resistance because this is genetic in nature. So in the next year, it will produce numerous weeds. Okay, from that mother plant, there will be more sister weeds will be formed. So all these weeds will be showing resistance to that particular herbicide. So after the herbicide application, then there will be no effect on those weed flora because they have already developed resistance, right? So here you can also find, suppose these are the initial weed flora and one plant showed resistance. Okay, then after we have applied the herbicide, some plant will get killed, but this plant, as it is showing resistance, it will survive. Then after 3-4 years, most of the weeds in that particular area, they will be resistant to that particular herbicide, and after few years, only resistance population you will find. Okay, so this is how herbicide resistance is very fertile. So percentage of resistant treated population, it goes on. If we cannot control that resistant population, it will go on increasing like exponentially. Suppose here we have applied in the first year one herbicide and it is controlling all the weeds in that particular area. Then second year also it is controlling all the weeds. Then third year it is also controlling all the weeds. Then there is some kind of mutation happens. Suppose in fourth year one weed survived. Then it produced some seeds. Okay, after flowering it produced some seeds. Those weeds germinated from those seeds of herbicide plant. They are capable to resist the herbicide application. So there will be resistance. Then in the next year, sixth year, then there are numerous seeds will be produced from those resistant weeds. So the herbicide resistance will go on increasing at an exponential rate, right? Next thing is types of herbicide resistance. There are several types of herbicide resistance you can find in the nature. That means first one is single herbicide resistance or simple resistance. Single herbicide resistance or simple resistance. Basically, when we apply one particular type of herbicide and it is being one weed is capable to resist to that particular herbicide, then that is called simple resistance. It is simply the resistance of a weed species due to continuous exposure to same herbicide. We are continuously applying, suppose let's say we are continuously applying 2,4-D in one particular area, right? So if that 2,4-D, it is being developed resistance, some weed being developed resistance to that particular 2,4-D, so they will escape. The application of herbicide will not affect that particular weed, right? So that is basically simple resistance or single resistance. Again, this resistance is of two types. One is complete or full resistance, one is partial resistance. In complete resistance, there will be no effect of that herbicide on that particular weed. Okay, resistant biotype are not at all affected by the herbicide used at recommended dose, remember. But in partial resistance, there will be inhibition of growth of that particular weed, but that will not be killed. That will not be fully controlled. That herbicide will not have the capacity to fully control that weed as it has partially developed resistance, right? Resistant biotype receive severe inhibition on their vegetative growth by the herbicide. However, they continue to grow insufficiently and reach to the flowering stage to produce certain seed. So they are not fully controlled and their growth stages will not hampered. Right, so that is partial resistance. Then cross resistance. Cross resistance means one herbicide, one weed, suppose it is showing resistance to one herbicide, okay, and that herbicide is having one mechanism of action. Mechanism of action means what? Mechanism of action means how that particular herbicide is acting. What is the actual phenomena that is being happening to control that particular weed by one herbicide. So that is the mechanism of action. If we are applying, suppose let's say we are applying one particular herbicide and it is inhibiting the photosynthesis, right? And this particular weed, it is becoming resistant to that particular herbicide. Then another herbicide, if it is also showing resistance, if this weed earlier it is showing resistance to one particular herbicide which is affecting the photosynthetic pathway. If this weed showed resistance to another herbicide which is also affecting photosynthesis, that means these both herbicides are having similar mechanism of action, then this is called cross resistance. Are you getting my point? One weed is there, it is showing resistance to a particular herbicide that is having one kind of mechanism of action, and if that particular herbicide after some years it developed resistance to another herbicide which is having similar mechanism of action, then that is called cross resistance. Let's listen the definition. When a weed species already resistant to a herbicide shows resistance to another herbicide of the same chemical class to which it has never been exposed, or it shows resistance to two or more herbicides due to single resistance mechanism, then that is called cross resistance. Okay. So here are some examples of cross resistance weed. So in wheat crop, Phalaris minor, it showed resistance to isoproteron. This is very common. You have to remember this. Phalaris minor, initially in wheat crop, Phalaris minor is one associated weed. So it initially developed resistance to isoproteron, and it developed later on. It also developed cross resistance to dichlorpropimethyl, phenoxapropiethyl, as well as sulfosulfuron. Right. So as this herbicide isoproteron, okay, Phalaris minor initially developed resistance for isoproteron, okay, and later on, as it developed resistance to isoproteron, we suggested, or scientists suggested to farmers, that you just go for alternate herbicides. So these were the alternate herbicides being applied to Phalaris minor for Phalaris minor control after it showed resistance to isoproteron. But gradually it developed resistance to all these herbicides, dichlorpropimethyl, phenoxapropiethyl, and sulfosulfuron. So this is one example of cross resistance. Then in maize and soybean crop, Sectaria faveri, that is one weed found in maize and soybean crop, it developed resistance to nicosulfuron, and later on it developed also resistance to imidazopyr. Okay, so this is one example of cross resistance. In rice, there is one weed that is called Cyperus deformis. That is one sage. Okay, Cyperus deformis. It showed resistance to sulfonylureas group. Okay, and it also showed resistance later on to bispyrimidax sodium, that is nomini gold, right? So this is another example of cross resistance. In wheat, another weed is also there, Lolium rigidum, that also developed resistance to dichlorpropymethylaldehyde and atrazine. Again, it developed resistance to chlorosulfuron and chlorotoluene. So these are some example of cross resistance. Are you getting my point? So cross resistance is basically if that particular weed is showing resistance to one particular herbicide, and again it is after some time again it is resistance to another herbicide of that particular group of chemical, then that is called cross resistance. Then there are two different types of cross resistance. One is target site based cross resistance, another is metabolic cross resistance. What is target site based cross resistance? If resistance to two or more herbicide of similar or dissimilar chemical group inhibiting the same target site. If the target site, as I was discussing, that mechanism of action of those particular herbicide, both the herbicide to which that particular weed is showing resistance, if the mechanism is similar, then that is, and if the target site is due to target site alteration. That means if particular herbicide is affecting at particular point or particular place of that particular weed, and that target site is altered due to some alteration in genetic level, then that is called target site based cross resistance. Like for example, triazine and triazinone, uracil, which inhibiting photosynthesis at pH2. So these herbicides like triazines, triazinone, what is their mechanism of action? They usually inhibit the photosynthesis and pH2. So there the target site is altered, due to which it showed a resistance. So this is called target site based cross resistance. Then metabolic cross resistance is the resistance to two or more herbicide of similar or dissimilar chemical group or their toxic metabolites due to similar metabolic degradative process. Okay. So another thing is, suppose one weed is showing resistance. It may be due to mutation or genetic level change in genetic constituent. And another thing is, if the metabolic rate is changed, suppose the metabolism is increased several times, so that particular herbicide will be metabolized, will be converted from toxic form to non-toxic form at a faster pace. So in that way also the degradation will be faster. Degradation of what? Degradation of the chemical, degradation of the herbicide, it will become faster. So that particular weed will become resistant. So if it is due to metabolic or degradative pathway, then that is called metabolic cross resistance. For example, Phalaris minor shows resistance to isoproteron and dichlormethyl due to metabolic cross resistance. Next thing is negative cross resistance or collateral sensitivity. Here remember, negative cross resistance, that means suppose we are applying one particular herbicide and this particular weed is resistant to that herbicide. Okay, and this is becoming extra sensitive to another herbicide of that same chemical group or same mechanism of action. Are you getting my point? Suppose one herbicide, one weed is becoming resistant to one particular group of herbicide, one particular herbicide. It will become extra sensitive to another herbicide. It is not becoming extra resistant, it is becoming extra sensitive to another herbicide. Then that is called negative cross resistance or collateral sensitivity. The mechanism by which an individual resistance to one herbicide or chemical family of herbicides shows higher or increased sensitivity or susceptibility to other herbicide than its natural wild type susceptible population due to changing in target enzyme. So target enzyme is here changed, so it is becoming resistant to one particular herbicide, but it is becoming very sensitive to another herbicide. Then that is called negative cross resistance or collateral sensitivity. For example, atrazine resistance biotype of Echinoclea crosscully. Suppose this Echinoclea crosscully is showing resistance to atrazine, but it is becoming more sensitive to fluazepop butyl, okay, and cethoxidine. So this is one example of negative cross resistance. Then multiple resistance. If one particular weed is resistant to one particular herbicide and it is showing resistance to another herbicide which is having different mechanism of action, right? Both of these herbicide is having different mechanism of action, and this weed is capable to resist to both these weeds, then that is called multiple resistance. A weed species show resistance to herbicide of different chemical class or family and having different mode of action or mechanism of action by two or more distinct resistance mechanism. Okay. For example, Lolium rigidum, it is one famous example. Lolium rigidum, it is showing multiple resistance to triazine, triazinon, phenylurease, sulfonylurease, imidazolinone, and glyphosate. So they are having different mechanism of action. Here glyphosate, it inhibits the EPSP synthase pathway or EPSP enzyme. Okay, glyphosate, the mechanism of action is inhibiting EPSP enzyme. Imidazolinone, the mechanism of action is inhibiting ALS enzyme, that is acetolactate synthase. Sulfonylurea, same. Then phenylurease group, it is inhibiting the PS2, okay, of photosynthesis. So these herbicides all are having different mechanism of action, and this particular weed, Lolium rigidum, it is capable to develop resist all these different herbicide having different mechanism of action. So this is one example of multiple resistance. Next thing is reverse resistance. That means if that particular herbicide is that particular weed is resistant to a particular herbicide, if this weed is resistant to particular herbicide, if we are stopping the application of this herbicide for 7 to 10 years, if we are stopping the application of or using the application of that particular herbicide as the weed has developed resistance, then after 7 to 10 years it will have sensitivity, it will develop sensitivity to that particular herbicide, then that is called reverse resistance, a phenomenon in which the weed biotype resistance to a herbicide falls susceptible to a very herbicide, to that particular herbicide, if it is becoming susceptible if it is not used for next 7 to 10 years instead of some alternative herbicide used to kill the resistance population. So initially it is resistance to that particular herbicide. Then we recommended the farmers to not go for application of that particular herbicide because it is already showing resistance. So after 10 years, if it is becoming again sensitive to that particular herbicide, then that is called reverse resistance. Then co-resistance or compound resistance, a phenomenon in which weed develops resistance to both mixing partner of herbicide mixture applied concurrently. So for example, Lolium rigidum resistance to amitrole and atrazine applied concomitantly, means if that is showing resistance to one herbicide mixture, okay, combinedly applied, so that is called co-resistance or compound resistance. This is one example. Next is mechanism of herbicide action. Okay, this was what is herbicide resistance and what are the different types of herbicide resistance. Next thing is mechanism of herbicide action, how herbicide resistance is happening. Okay, so there are various mechanisms. First one is exclusionary mechanism. These mechanisms exclude herbicide molecule from the site of action in plant where they actually induce the toxicity. So basically, due to some mechanism, that particular herbicide molecule cannot reach to the site of action. That is called exclusionary mechanism. For example, differential herbicide uptake. So if that plant, the uptake of herbicide, the translocation of herbicide or absorption of herbicide is inhibited, then that will show, suppose this is one weed and we have applied herbicide over it. Okay, if there is certain changes in the leaf structure or leaf composition and that particular herbicide is not penetrating inside the leaf, so it will not translocate and it cannot move to the site of action. So due to this process, due to differential herbicide uptake or differential herbicide absorption, if the resistance mechanism is developed, then that is called exclusionary mechanism. Another form of exclusionary mechanism is differential translocation, right? If the translocation of the herbicide is less in the resistance biotype, then that is another form of exclusionary mechanism. So due to, if the translocation is very slow, becoming very slow, then that herbicide, it will take longer time for that herbicide to reach to the site of action. Then there will be herbicide resistance will be developed. For example, Lolium rigidum, this is resistance biotype. So less translocation of chlorosulfuron when compared with susceptible one. Okay. So these are some kind of exclusionary mechanism. Then sequestration of compartmentation. If that particular herbicide is sequestered with some biomolecule or combined with some biomolecule, then it cannot be translocated and available for the site of action to act properly, right? So if it is binding with some biomolecule inside the plant system, inside the weed system, so it cannot reach to the site of action and it cannot kill that particular weed. So that is another example of like mechanism of resistance. Then enhanced metabolism or degradation. We have already discussed, if the metabolic rate is higher, if it is becoming higher, then it will degrade that particular chemical quickly. So the action will be very limited. So next is, first one is exclusionary principle. Then target site mechanism of resistance. If there is any alteration, if there is any change in the target site, okay, where the herbicide actually acting at the target site, that if there is any change in that particular target site, then that is called target site mechanism of resistance. First one is altered site of action or target site alteration. If there is genetic change due to which the target site is changed or the target site is blocked, then herbicide molecule where it will attach and where it will act, it cannot act properly, right? So if it refers to the modification or alteration to the binding site of the herbicide due to some genetic changes, due to genetic change in the binding site, there is some alteration. So this becoming resistance, okay, because herbicide molecule cannot get bind properly or cannot be fit properly in that action site, so it will remain unaffected, right? So that is called altered site of action. For example, lactuca sativa biotype resistance to sulfonylurea have ALS enzyme modified in such a way that this herbicide can no longer bind and inactivate it. For example, this is one enzyme present in that particular weed, okay, and if that target site is clear, if it is not altered, so that herbicide molecule, it can move and it can attach properly with this target site, okay. If there is any change, okay, so for example, if there is certain change in this target site, for example, here it is like this structure and here it is triangular structure. So here herbicide is having same chemical structure, so it cannot bind properly, okay. This is the target site, this is one suppose this is the structure of the target site, okay, and there is genetic change happen or mutation happen and the target site become like this, okay, but the herbicide that is coming to attack it, that is having this type of structure, so it can fit properly here, so it is earlier showing susceptible, showing effect on that particular weed, but after the change in target site, it cannot attach here, okay, completely, so it cannot control that weed. So this is target site alteration due to some genetic change. Then target site overproduction. Then another mechanism is overproduction of target site. That means if suppose some limited number of target site is present and we have applied herbicide, okay, so it will automatically fill that target site and it will kill the weed. If this target site produce in a large number and we have applied limited quantity of herbicide, so it can control one or two or some target site, it can bind and it can control partially that weed, but other target sites are yet blanked, okay. So due to overproduction of target site, the resistance mechanism may develop. So that is called overproduction of target site or site of action. In this mechanism, the site of action or target enzyme is produced in excess quantity. As a result, the herbicide at a normal rate of application is not able to inactivate the total enzyme produced. For example, Allopecurus myosuroid, that is called blackgrass, is resistant due to increased activity of glutathione S transferase. Clear? So these are certain mechanism of herbicide action, okay. Then this is one example, like initial year there are less resistance weed present, so there is more grain yield. Then later on, due to increase in resistant weeds, there will be more infestation and the herbicide is incapable to control those weeds, so there will be more yield loss. So this is one example of. Yes. Then management of herbicide resistant weed. How we can manage this herbicide resistant weed, or if any in particular area there is development of herbicide, how we can control that particular resistant weed, or how we can manage that there. So first thing is, where resistance is already coped up, that means resistance is already being developed in that particular area. What we need to do? First thing is farmers awareness. We must let the farmer know that this particular weed has already becoming resistant to that particular herbicide, so you just stop using that herbicide. Okay, that is farmers awareness and training and participatory approach. You must visit to village and you just aware the farmer that this particular weed is developing herbicide and developed herbicide resistance to that particular herbicide, so you just apply any alternative herbicide, right? Then second thing is abandonment of herbicide to which weed showing resistance in rapidly from that particular moment you just stop using that herbicide, and we should recommend the farmers to not use that herbicide. Okay. Then stop sale or purchase of that particular herbicide. Okay, so we should remove that herbicide from the market to control the herbicide resistance, to control the resistance to spread, right? Then prevention against spread of resistance weed. We should clean those resistance weed as quick as possible. We should not let those resistance weed to flower and produce seed, or dissemination of seed. We should not let it happen. Okay, if once that resistance weed produce seed, because weeds produce numerous number of seeds, like lakhs, one weed can produce lakhs or more than that seeds. So that seed, weed that will germinate from that seed which has already been resistant to a particular herbicide, so that weed also show resistance to that particular herbicide. So we should not let the weed to flower and disseminate the seed. So this is another thing. So we must clean the tillage implement or harvesting equipment, and we must rock out that particular weed. Next is cultural approaches. There are different cultural approaches we have already read in the control section, like we should go competitive and high yielding varieties, then pure and certified seeds. These are some preventive approach, tail seedbed technique, then closure, row spacing, proper time, method and rate of sowing, crop rotation. So these are cultural weed control measures. Same measure you should follow to prevent the resistance weed growth. Then mechanical and manual approaches like tillage practices, soil solarization and harrowing we have already discussed. Then herbicide management. Use alternate herbicide at appropriate time and dose. And we should go for herbicide mixture, those which are having different mode of action. We should use that. Next thing is herbicide rotation. We must rotate the herbicide. Okay, we should not go for a single herbicide application year by year. Okay, we should rotate the herbicide so that the chance of herbicide resistance development will be minimized, right? Then we should go for integrated weed management. We should not depend only on herbicide. Rather, we should go for combination of different aspect of herbicide weed control, like preventive approach, cultural approach, then mechanical approach, then biological approach, then chemical approach. All this mixed approach of herbicide, suitable combination of the weed control methods we should go for. Then monitoring resistance weed population towards cross resistance or multiple resistance. We should monitor the resistance weed population, whether if we are going for application of any alternate herbicide and it is showing any cross resistance to that alternate herbicide or not, or multiple resistance to that alternate herbicide or not. We should study that or monitor that. So this is how herbicide resistance can be managed. Okay, so what should be the role of pesticide industry? It should develop low risk novel broad spectrum herbicide. Broad spectrum herbicide means the herbicide should be capable enough to control a wider range of weeds like grassy weeds, broadleaf weed and sedges. That is called broad spectrum herbicide, and it should be low risk. That means it should have higher LD50 value. Then it should develop, the herbicide company or pesticide company, it should develop the cost effective herbicide mixture Then it should properly level, or you should probably give the guideline for herbicide uses, because farmers, what they do, instead of applying the recommended dose, they are going for higher dose of application. So in that case, chance of herbicide development or resistance development is higher. Then monitoring the product at the factory and in the crop field as well by the personnel, research and development personnel. Then academic and institution, what they can do, they can educate or train the users of that herbicide. They can do some training programs to aware the farmers about the herbicide resistance. Then monitoring herbicide use and resistance biotype, we can do that. Then conduct research and extension services regarding resistance management. This is the role of academics or institutions. Then government agencies, what is their role in managing the herbicide resistance? It should regulate the herbicide. Okay, it should, and the herbicide must be registered. Then funding research and extension activities regarding herbicide resistance. Government should promote the funding for research on resistance, herbicide resistance. Then promote integrated weed management. Government should make some policies to go for integrated weed management rather than single use of herbicide, only use of herbicide. Then what is the role of user end or farmer's end? It should go for crop cultivation and adoption of IWM, that is integrated weed management. It should go for herbicide rotation as well as herbicide mixture. Then farmers should maintain the year-wise data that which herbicide he is using and which type of weeds is being grown in that particular area. Then use of competitive varieties and crop rotation, that is basically we have already covered in cultural aspects of weed control. Then alternate use of resistance weed. Okay, alteration use of resistance weed. So these are different like pesticide industry, what is their role. Then academic or institution like colleges, universities, what is their role. Then government agencies, what is their role, and farmers or end users, what should be their role in management of herbicide resistance weeds. Okay, next thing is where resistance, okay, this can be done where resistance has already been developed. So in that place where resistance has not yet developed, what we must do to prevent the herbicide resistance, okay, to avoid the resistance development, what we can do? First one is farmers awareness and training. So we must aware the farmer that you should go for integrated weed management, you should rotate the herbicide regularly. Then you should keep track on which herbicide you are using or which type of weeds are coming up in your field. That we should that we can train the farmers. Then monitoring change in weed population towards resistance, if any, while using herbicide across the year. So you should track that after repeated application of herbicide, what is the change in weed flora in that particular area. Then herbicide management and integrated weed management, this we can follow to prevent the herbicide resistance in a particular area. Then what should be the ideal IWM practice? That means what should be the ideal integrated weed management practice. So right now the scenario is like in integrated weed management, mechanical approach, suppose this one, and this middle one is cultural approach. So this fraction is very minimal right now, and we are mostly dependent on herbicide for the control of weeds. The ideal should be like equal division, herbicide, mechanical as well as cultural. Their use should be equal. That means we should not depend more on herbicide approach. We should go for integrated approach. That doesn't mean this, that means this. That means we should equally emphasize the mechanical approach as well as cultural approach as well as herbicide approach for our weed management. That is one optimal integrated weed management practice. Next thing is, what is the current status? That means this is not current, actually this is of 2016. So you can see here, during 1970 the first report of herbicide resistance weed, that was already we have discussed, that Senecio vulgaris, that developed resistance to atrazine weed. So after that, here the usage of herbicide is increased, and the resistance also developed after 1975 or 1980. The resistance development has like it is exponential, right? And mostly the herbicide resistance weed, they showed resistance to ALS inhibitor group, followed by the ALS inhibitor group followed by the PS2 inhibitor group. Okay, so here you can see this is ALS inhibitor group. Okay, then followed by the PS2 inhibitor group, followed by SCCS inhibitor group, right? Next thing is okay, so these are some reports you can go through if you have time. Then like number of resistance biotype, here you can find it is up to 2000. So up to 2000, it is also reported that more than 300 biotypes has already been reported as showing resistance to different herbicides. So number of herbicide resistance species by crop, we can see in wheat, more number of weeds has already shown resistance to different herbicides. So the number is 73, and this data is also from 2016, followed by corn, because wheat is grown almost in a maximum area of cultivation is covered by wheat, followed by corn, followed by rice. So the herbicide usage is also likewise. So the maximum development of herbicide resistance weed is found in wheat, followed by corn, and followed by rice. Next is number of herbicide resistance weed by family. So most of the weeds, they showed resistance, they developed resistance, that is from Poaceae family, followed by Asteraceae family, followed by Brassicaceae family. Next is increase in glyphosate resistance weed. So first report of glyphosate resistance, it was by Regi raygrass during 1995 to 2000, near about 96. So after that, goosegrass showed resistance to glyphosate, and likewise the list is increased, as you can see here. And right now there are more than 35 to 40 species they are showing resistance to glyphosate. So status of herbicide resistance in India, the first report was Phalaris minor. We were already discussing that it showed resistance to the isoproteron. Initially, the Phalaris minor, that is one associated weed in wheat plant, that showed resistance to isoproteron, then followed by diclofop-methyl, phenoxaprop, chlorinafop, sulfosulfuron. So many herbicide it shows resistance, right? And later on, so this was isoproteron. Then later on, little-seed canary grass, that is Phalaris minor. So basically, Phalaris minor is so resistance to this one is the report by Samundar Singh in January 2014, and another report by Rajendra Chokar in January 2014. It also reported that Phalaris minor has shown resistance to SCCS inhibiting herbicide as well as ALS inhibiting herbicide. Okay, so herbicide resistance weed, this is the list. So you can it is easier to follow up. So these two and these weeds actually they showed resistance to different herbicides, and here the years are also mentioned and what is the site of action. So Phalaris minor developed resistance. The report has been like 1991, 94, 2006, even 2013 to different site of action like PS2 inhibitor, urease and amide, SCCS inhibitor, then multiple resistance to SCCS, ALS and PS2 inhibitor, then ALS inhibitor. Then Rumex dentatus, it also showed resistance in 2014 to ALS inhibitor herbicides. Next is, what is the basic difference between herbicide resistance and herbicide tolerance? Herbicide resistance, remember, this is due to the genetic change. That means either due to mutation or due to any alteration in the genetic level. So it is hereditary in nature. Herbicide resistance is hereditary in nature, and it is due to change in genetic constituent. Whereas the tolerance, that is basically environmentally it is tolerated. That is actually tolerated from its very beginning. That means suppose resistance is developed gradually, but tolerance, it is from its beginning. That means it is not genetically controlled, right? So let's read it out. Herbicide resistance is the inherited ability. That means it is hereditary. That means it is going from generation to generation, moving from generation to generation. ability of plant to survive and reproduce following exposure to a dose of herbicide normally lethal to the wild type. In a plant, resistance may be naturally occurring or induced by such techniques or genetic engineering or selection of various variants produced by tissue culture or mutagenesis. It can be naturally occurring, it can be produced in the laboratory also. Whereas herbicide tolerance is the inherited ability of a species to survive and reproduce after herbicide treatment. This implies that there is no selection or genetic manipulation to make the plant tolerant. It is naturally tolerant. So this is the basic difference between herbicide resistance and tolerance, remember. So how to calculate the herbicide resistance? There are two different terms. One is resistance index, another one is resistance factor. In case of insecticide, if any insect is developing resistance to any any insecticide, then we use mostly resistance index, okay, that is represented by LD50 value. And in case of weed control, we use the term resistance factor, that is represented as GR50 value. So what is resistance index? That means LD50 value of resistant population, okay, divided by LD50 value of susceptible population. So as we are using resistance index in case of insects, that means what should be the dose of the insecticide to control 50% of the resistance insect, divided by what should be the dose of herbicide or insecticide, because as we are using resistance index, so what should be the dose of insecticide that can be controlled 50% of susceptible population. So this is basically resistance index. Then what is resistance factor? That is the growth reduction 50, means 50% weed population. If we want to reduce the growth of 50% weed population of the resistance population, what should be the dose, divided by dose of herbicide for 50% growth reduction in susceptible population. So this is a resistance factor. So clear what is resistance index and what is resistance factor. It is continuously coming in exam, so you must remember this. Next is let's say, so in our conclusion we can see that what is resistance, we just gone through what is resistance. Then there are different types of resistance, like simple, we discussed simple resistance, cross resistance, multiple cross resistance, then reverse resistance or inverse resistance. So there are different types of resistance. We have already discussed how we can manage the herbicide before the resistance development and after the resistance development. What are the precautionary measures we should take, follow up to prevent the resistance development. Then we discussed what is herbicide resistance, what is herbicide tolerance. Then what are the weeds that are being resistant to different herbicide in India. Then we discussed what is resistance index and what is resistance factor. So if you have any doubt regarding this topic, then you can let me know in the comment section so that we can discuss further. See you in the next lecture. Till then, bye bye. Have a